sense on here, I promise. All right. So here we go. I handed out a little just schematic overview like this. I'm a simple guy, I'm simple-minded, and this is simple for me to see because kind of this is our roadmap of kind of what we're going to be talking about today. This breaks down the nervous system if you go kind of left to right from, you know, the nervous system itself as a whole and then some of the major parts, and then it keeps further breaking it down just so you kind of know what is a subcomponent of another part of the nervous system. And this is something when I'm studying or something, I'll just have it sitting next to me. So I'm like, now what was that a part of? And what is that basal ganglia, that globus pallidus, or that whatever is a subcomponent of? And that helps me to just kind of keep that stuff straight. So use it however you'd like to. So basic components, nervous system. What have you, most of you guys, what are your undergrads studies in? Is it a pretty big variety? Some exercise science, exercise phys, what? Pre-dentist? Nursing, anything like that? Biology? So most of you like at least had a class that talked about the nervous system at all? Okay, so anyway, we're going to assume you know nothing, so that's okay. If you haven't had it at all, that's okay. So we're going to, 4 basic components. Neurons, neuroglial cells, okay, that's the most, lowest component or the most, cellular level of the components of the nervous system. Then we have this thing called the central nervous system, which is made up of neurons and neuroglial cells. A peripheral nervous system that is also made up of neurons. And then the autonomic nervous system, which is also made up of neurons that are both contained in the central and peripheral nervous system. So we're going to go through each of those right now, those 4 major components first to start those out. So neurons, this is what is the thing that allows our nervous system to work, to communicate with each other, to send signals from one place to another. It is the basic structural functional unit of the nervous system. If a neuron tends to convey information or brings information from the periphery towards the central nervous system, or from one structure within the central nervous system towards another structure, they typically call that an afferent neuron. And I think of the A as kind of like acquire, acquiring information. Afferent, acquire, that's how I remember bringing it in, acquiring, so afferent. If a neuron is taking an action potential or a signal and sending it away from a structure or from the central nervous system out to the periphery, like to a muscle, if that signal is going that way, it's exiting, so it's an efferent. So E, exit, efferent. That's kind of how I remember. It's an efferent type of neuron. If it's only communicating with between neurons, okay, so a neuron, typically an afferent or an efferent is communicating with some other structure, a gland, a muscle, or bringing in information from a receptor, some kind of thing like that. If you have a neuron that's only communicating with other neurons, those are called interneurons. The ones that these, and most, 90% of our neurons are interneurons, but we'll talk about, when we talk about motor and sensory pathways, we're talking, those are the afferent, efferent neurons, which actually make up a small percentage of our nervous system, but it's those interneurons that make up 90%. And that's where all the real processing comes in. You bring in information, it gets processed, and then you send information out, but the processing is all the interneurons, okay, and their relationship and communication. The other part of the cellular matrix or part of the nervous system, in addition to the neurons, which have the action potentials and can actually fire and depolarize and all that kind of stuff and communicate, is neuroglial cells. Okay, so neuroglial cells. Glia is glue. They're like kind of the glue or the sticky stuff that kind of holds the nervous system together. Okay, glial cells are non-neuronal cells that I would put an asterisk, or they do not convey information as a neuron would, but there's actually some really interesting new work showing that we actually probably do, glial cells actually do have some communication ability. It just doesn't, they're not sending action potentials. They communicate by moving certain molecules from one to another and so forth, but it's not in the traditional sense of an action potential in the nervous system or a nerve depolarizing. Glial cells are a little different, but they may actually shape and help to shape information and how it's processed. May mainly insulate. They provide actual insulation. They're the cells that actually they wrap around the axons of your neurons, the myelin that insulates it like a wire, so it sends a signal more quickly and doesn't get lost. They nourish they provide some nourishment. Okay, they support and they protect. They actually provide that structure The glial cells form the meninges, which are the coverings of your nervous system, which actually provide some protection. So that's what they do. All right, central nervous system, the big one, okay? Central nervous system. Traditionally, people talk about 7 main parts, and this varies from book to book, but we're gonna stick with this: 7 main parts of the central nervous system, okay, which is made up of neurons and glial cells, okay? We have these main structures. We have the cerebral hemispheres. The big cerebral hemispheres you typically think of when somebody says the brain. You picture a brain with all the wrinkles on it. That's what we're talking about, these cerebral hemispheres. Down deep inside, right in the middle of the brain, or in the heart of the brain, is a structure called the diencephalon. Di, to, encephalon within the cephalic structures, kind of deep in the brain. The diencephalon is typically, we're talking about 2 structures, the thalamus and the hypothalamus. That's why it's the diencephalon. Okay, it's these 2 structures together inside your cerebrum. Okay. You have then your brainstem, which is really part of your brain proper. And the brainstem has 3 parts. It has a midbrain, which is actually kind of up in the middle of your brain when you look at it, cross-section. We'll show you that in a second. You have the pons, which is the next thing below the midbrain. And you have the medulla, which is below the pons. Okay? Sitting off the back of that brainstem is the cerebellum. And then finally, at the medulla, once you exit the foramen magnum, that hole in the bottom of your skull, now you're called the spinal cord. Okay, so that's the 7 main parts, and you can see them here. And I have my little drawing tool, so I can draw stuff on here if you want to. If I can find my tool here. Hopefully it works. Yep. So the cerebral hemispheres, this whole area here, this lighter, whatever that is, tan or something like that, the wrinkly stuff. Right now, what you're looking at here is you're looking at a cross-section, a sagittal section right through the middle of my skull here. So you're looking at the medial or inner surface of that. That's a right hemisphere, and you're looking at the medial surface being cut right down between this called the cerebral longitudinal fissure, and we're looking at the medial surface of it, okay? And then the cerebral hemispheres. Here's your diencephalon, is this pink structure in here. It's divided into the thalamus is really just this region here, then the hypothalamus is all this stuff just below it, okay, a little bit in front of it and below it, the hypothalamus. Together they're the diencephalon. And then here we have the midbrain. So we're in the brainstem now. The midbrain is right here. And you can see it's kind of in the middle of the brain when you look at that. And then this guy, the little bit larger structure here is the pons. The front part here, this bulging part's called the basilar pons. And then we have our medulla. And then here's our foramen magnum, the hole in the bottom of our skull, and then that's our signification that we're now in the spinal cord. And then sitting off the back here, we have this structure called the cerebellum off the back of the brainstem. So those are 7 main parts. You guys good with that? Hanging in there so far? Probably going to take just 1 break today, but if you have to get up, go take a break and get something, go use the restroom. It's not going to bother me. Feel free. So. Peripheral nervous system is the next one here. So we talked about the neurons and neuroglial cells, central nervous system. Now I'm peripheral nervous system. This is all parts of the nervous system not encased in the skull or the vertebral column. Okay, so once the nerves come out of the skull or the brainstem or the spinal cord, they're peripheral nervous system. Okay, what we're talking about here is the cranial nerves and the spinal nerves. So the cranial nerves are the nerves, the peripheral nerves that exit from your brain and brainstem. Okay, they come out. They're kind of like spinal nerves, but they're just coming out of the cranial area. Okay, so don't get overwhelmed by cranial nerves. They're very similar in function as your spinal nerves, but they're just going to your head, face, neck. But they also serve some special functions like vision, some autonomic nervous system function, and those kind of things, hearing, and those kind of things. So they have some specialized function. You have 31 pairs of spinal nerves. You have 12 cranial nerves. You can add that, 12 pairs of cranial nerves, so they come in pairs, okay? Because they're going to each side of your body. So in this picture, everything in the grayish blue is the central nervous system, and all the yellow stuff. Everything in yellow is peripheral nervous system. So in those peripheral nerves, most of them all contain, until you get to the very end, When they're coming out of the spinal cord, okay, especially we're talking about spinal nerves coming out of the spinal cord, they're going to have usually some motor, some sensory, and some autonomic. Okay, so they're going to have all three of those in it. It's not till they finally get to their very final destination when they break off to just supply a muscle or they're just coming from a receptor. But once they get into that big bundle of nerves, you've got a mixture. Okay, so especially as you get closer to where they're going to go in or out of the spinal cord. Okay. Now the autonomic nervous system is it is part of the central and peripheral nervous systems, but it has components in both, okay? So the autonomic nervous system is that part of our nervous system that's mainly for that regulation of just basic body functions, okay, that we don't think about. It's not under conscious control, okay? So this is regulating blood flow, temperature, homeostasis is what we're talking about here. And so you have that sympathetic part, okay, the fight or flight response. You know, it's going to ramp up metabolism, ramp up breathing, ramp up heart rate, turn things up most of the time, okay? That's known also if you hear them talk about the thoracolumbar division of the autonomic nervous system is simply the same as saying the sympathetic nervous system. So the thoracolumbar division of the autonomic nervous system is because your sympathetic nerves come out in that thoracolumbar region. The nerves are going to go tell your body to ramp up function, ramp up heart rate, ramp up breathing, and they're coming out between T1 and L2. That's why they call it the thoracolumbar part of it, okay? Then the parasympathetic nervous system is also sometimes known as the need and feed. This is going to be for storing energy, reducing heart rate, reducing breathing rates, those kind of things, altering blood flow to certain parts of our body depending on need. The parasympathetic nervous system is very important for that. It's also called the craniosacral division because part of the nerves that come out are cranial nerves that come out to supply a lot of your parasympathetic control, especially vagus nerve, cranial nerve number 10. It controls about 70% of your parasympathetic control of your body, that one big vagus nerve that goes down, drops down into your rest of your body from your cranial area, from the brainstem down to supply the parasympathetic control of a lot of your functions in the body. Then you also have some that come out in the sacral region, especially to control genital areas, lower bowel, bladder function, and so forth. So parasympathetic control of those comes out of a few sacral spinal nerves. So that's why it's called the craniosacral, because it's got cranial part and a sacral part, okay? It's also called the parasympathetic because it's on each side of the sympathetic's in the middle where it comes out. The sympathetic nerves come out thoracolumbar region, and the parasympathetic is on top and bottom of it, craniosacral, okay? So that's kind of mainly homeostasis. These are both sympathetic and parasympathetic primarily innervate smooth muscle, cardiac muscle, and secretory glands, okay? Unconscious control of body functions. This is just a schematic showing kind of how that works. So you have, you know, on the left side, sympathetic. Here's the thoracolumbar region. And so you do have higher centers of control in the brain, and actually mainly in the brainstem, that influence these different levels of the spinal cord and the amount of sympathetic outflow coming out of those, but they all exit out here in those thoracolumbar region, okay, is where the nerves go out to your body to control that. Then here's some parasympathetic control here on each side, and so this is the cranial part, and then down here's the sacral part. So you can see this is vagus nerve, and you see how vagus nerve comes down and just innervates a lot of your organs to do parasympathetic control of the organs through the vagus nerve. Okay, we have a few other cranial nerves also contain some parasympathetic fibers. The facial nerve does, the glossopharyngeal nerve. We're not going to get into that kind of detail today, but that's cranial, sacral, or parasympathetic nervous system. Those are where those nerves are coming out to, again, innervate organs. All right, that's the big overview of the four main parts of the nervous system we talked about. So now we're going to talk a little bit about language here of neuroanatomy, just to help you, because if you know a few key things, it can help you when you're trying to put together terms. When you're reading something about a certain part or region, you're like, oh, that term generally means this, so you can kind of infer, that if you hear the term tubercle, that's going to be, you know, it's a bump. A tubercle is a bump on the surface of a nervous system structure. Okay. Let's get oriented to location here. Do you guys know about, like, you know, body orientation, anterior, posterior, inferior, superior, that kind of stuff? A little bit of a quirk in the nervous system. Okay. The axis that runs through the central nervous system, it's running straight up and down, and it runs along with the body's axis when it's in the brainstem and the spinal cord. So when you're talking about structures in the brainstem or the spinal cord, you say something's anterior, posterior to it, you know, or superior or inferior or rostral or caudal. It's the same as in your body, okay? Same orientation as the rest of your body would be, okay? Your trunk and those kind of things. But when you get into the cerebrum right here, it actually changes. The axis tilts like this, because there's actually ways, as your brain is formed, everything's in alignment. And like reptiles, everything is lined up. Their spinal cord, brain, and brainstem, it's all lined up. But in humans, when we go to be able to look forward, and our eyes have to look forward, so we're not on all fours, we're on upright, there's this thing called the cervical flexure. So your brain actually flexes over the top of your brainstem and orients this way. So the axis runs this way. So all you have to know about this is if you're asked a question about where a certain structure is in relationship to another structure, you have to just ask yourself, am I in the cerebrum or am I in the brainstem or spinal cord? Because that will change where something might be related to each other, the terminology used. So if you're in the cerebrum, you know, you'll see that rostral. You guys know what rostral and caudal is? Rostral means beak, or towards the beak, okay? And caudal means towards the tail, okay? If you are in the brainstem and spinal cord, caudal and inferior would be synonymous. So if somebody asks you, you know, so the level of C8 is caudal or inferior to C7, right? So the C8 nerve roots are below, and it's caudal or inferior if you're in the spinal cord, okay? If you extend this up here like this, if we extend that up, here's superior, and this would also be rostral if we're staying on this vertical axis here. And so C7 is, in the spinal cord, is superior or rostral to C8. Okay, so C7 is above it. But if you get into the brain, and we talk about structures in the brain, so let's say the frontal lobe here. Let me erase some of this stuff to get some of the confusion out of here. Here's the frontal lobe, and here's the occipital lobe back here. So in this case, we'd say the frontal lobe is rostral, because this has changed, this axis, or anterior to the occipital lobe. So in the cerebrum, rostral and anterior are synonymous. Does that make sense? However, in the brainstem and the spinal cord, rostral and superior are synonymous. That terminology is synonymous. So just because of that change in axis. And here, and then in the cerebrum, the occipital lobe would be caudal or posterior. So these are synonymous. Caudal and posterior synonymous in the cerebrum. However, in the spinal cord and brainstem, caudal and inferior synonymous. So I could use either one of those terms to describe locations. You just got to make sure you know what region you're in. And if you kind of have this picture in your head, it doesn't, I'm making it harder than it probably needs to be if you just kind of have a picture like when you're thinking about it. Because if somebody's going to ask you a question about neuroanatomy, these have to describe where it is in relationship to another object. And so it's important to kind of know that. So, and you can see here, so see how ventral, the front of our body is also the ventral surface, the dorsum back here is the back. That's in the relationship to the body or to the spinal cord and the brainstem. But when you get in the cerebrum, dorsal is going upwards and ventral is down or inferior. So you just have to kind of be aware of that. So when you're reading books about where things are, just say, ask yourself if you're in the cerebrum or if you're in the brainstem or spinal cord. Anatomical planes, okay, so when we cut through specimens and divide them up, you want to know about these different planes. We have a coronal plane, which is also known as the frontal planes, and I'll show you a picture here. Sagittal, and we have a median or mid-sagittal or parasagittal sections we can make through these structures, and then the horizontal or transverse, okay? So these are the planes here. So here's the coronal plane. That's as if somebody dropped a sheet of glass this way or right through me this way, and you're looking straight on this way. That's a coronal plane kind of cut, okay? Then the sagittal cut is right down the middle this way. So sagittal plane is you drop a sheet of glass or a guillotine or whatever you want to think, right down through the center this way, and you're cutting this way and you're looking at it this way to this way. That's a sagittal plane. Then horizontal plane cut is like this, straight through this way, through whatever section of the brain, cerebrum, cerebellum, brainstem, that's a horizontal, okay? These are central, these are like mid-coronal, mid-horizontal, and mid-sagittal cuts right there, right through the center of the brain. And anything off to the side are called like parasagittal, paracoronal, parahorizontal. You can kind of, you can go off to the side and start making slices every centimeter, every millimeter and slice through the brain in different sections. We're going to just focus on some of these major mid-sagittal, mid-coronal, mid-horizontal cuts. We're not going to do much more than that today. We just don't have time, okay? So here's our horizontal section. Okay, so that's cut right like this, and we're looking down at the lower part of the brain there. The top part of the brain's been cut away and taken off. Okay, here's a mid-coronal section. So we're looking, the front part of the brain's been taken away, and we're just looking at the back kind of half, but we're looking at that mid-coronal section. This is a mid-sagittal section. So this is where the brain has been cut right down the middle between the cerebral longitudinal fissures. This is what the brains you guys will be looking at are cut mid-sagittally. So you can look at the outside and kind of the inside of the brain. This would be a horizontal section through the brainstem. Okay, that's a horizontal section, that bottom picture, through the brainstem. Still on terminology here, okay? If you hear any of these terms in relationship to any structure, okay, if these terms are tacked on to the end of a structure's name, all you're thinking here is this means they're talking about some kind of a pathway or group of nerve fibers or axons, either running in that structure or beneath that structure, okay? Because when we get to some of the surface anatomy, we'll talk about some of these, and like, oh, that's just a group of axons running below that because it says it's a fasciculus, okay? So if you hear terms like capsule, radiation, lemniscus, tract, fasciculus, funiculus, a nerve, a commissure, all those terms are associated with groups of axons, okay? Either the group of axon itself or something like a structure like the surface structure, and below that thing is going to be a group of axons running some kind of pathway or connection between axons. Skip. If you hear these terms associated with a structure tacked onto the end of the name of something, a ganglia, a lamina, a body, or a nucleus, okay? We're not talking about the nucleus of a cell. When we get into the central nervous system and they talk about a nucleus, we're talking about a group of cell bodies together in the central nervous system. In the peripheral nervous system, the group of cell bodies associated with neurons tends to be called a ganglia, okay? There is one exception to ganglia used in the central nervous system called the basal ganglia, although they're trying to change it to basal nuclei because you should be consistent in your terminology. I have a book this thick that it's called Alternative Neurologic Terms. So for every neurologic term, they give you all the alternative terms, which every term uses about 20 other alternative terms that are used. So there really is about coming up with common communication, and even professions like PT, PA, how do you communicate unless you're all talking the same language? So when we get to the surface anatomy, things that are called like furrows or depressions, so hollowed out areas in your nervous system, spaces, grooves, whatever. If you hear sulci, fissure, fossa, okay? And you see that also on bones, right? So they use that same terminology on some bones. Same thing with these bumps and ridges. If you see bumps and ridges, they're called peduncles, gyruses, brachiums, or tubercles. are some of the common terms you see associated with that. And then we get into like layers of the nervous system. Like, you know, a lot of our nervous system has different layers to it within the spinal cord, the surface of our brain. Things like lamina, cortex, tectum, tegmentum. These are layers within the nervous system. All right, here we go. Going into basic anatomy. Any questions so far? Feel everybody's on the same page? You guys look like you're nodding pretty well. So now we're going to get into kind of this sheet right here. Now we're getting in a little deeper into the cerebrum area here. So we're going to go through and just talk about major function and parts. Okay, major function and parts, and then we'll eventually get to see this stuff in pictures and then on the brains. What does the cerebral hemispheres do? The thing we typically think of when you think of the brain. It does this: processes sensory information, motor, memory, reasoning, language, intelligence, personality, emotions. So it's really important. It makes you who you are. That's what determines who you are, for better or for worse. So cerebral hemispheres. It has 4 primary lobes: a frontal lobe, parietal lobe, temporal lobe, and occipital lobe. Okay, so that's what you commonly read. There is another lobe that, if you pull part of the brain, and we'll show you how to do that, you can look a little deeper. There's a thing called an insular lobe or a limbic lobe that some textbooks refer to. Okay, it's more autonomic function, and limbic or emotional kind of structures deep inside. Okay. We have a longitudinal fissure that divides the 2 cerebral hemispheres. So there's that big groove right down the center of your brain. That's the cerebral longitudinal fissure. It's a big, deep fissure. You have a corpus callosum. If you go down, you start digging down into that cerebral longitudinal fissure, you won't get very far. You might get an inch, maybe a little around an inch or so. Then you'll hit a structure that's kind of this nice, glistening white. If you pull it apart, this really white, shiny structure that is the corpus callosum. Okay, it's a commissural fiber. It's these axons that are connecting both hemispheres. The corresponding areas of your frontal lobe, parietal lobe, occipital lobes are all being connected from side to side, left to right, by this thing called the corpus callosum. So you can talk one side of your brain to the other. Okay? And then on the very surface of your cerebrum, okay, is you just have that little layer of gray matter. So when we cut through this, you don't see a section on these brains. You got to do either a horizontal section or a frontal section. You can do sagittal, I guess, but you got to be able to, so you can see the white matter versus gray matter. So we're talking about the gray matter is this little tiny, millimeter or so thick that actually has 6 different layers to it, but it's still only like a millimeter thick. That's where all the magic happens as far as the processing. That's where your cell bodies are. For your neurons, that's where a lot of communication is happening at that level. And then just below that, most of the rest of the meat of your brain is all white matter. It's all the axons that are running from the cortex down to and from the cortex. That's the axons or the white matter. Okay, so in the cerebrum, gray matter's on the outside, white matter's on the inside. Okay, cell bodies on the outside, white matter on the inside. Mostly, there's a few exceptions. But when you get down into the spinal cord, it's the opposite. The gray matter's in the middle, the white matter's on the outside. Don't ask me why it happens that way, but that's just the way it is. Okay. We do have a few important structures deep in the cerebral hemispheres. One we already mentioned, the diencephalon, which is just the thalamus and the hypothalamus together. Okay. But then we have these structures called the basal ganglia or the basal nuclei, which are a collection of structures, okay? So it's plural, basal ganglia or nuclei. There's multiple structures that make up. They're functionally associated with each other. Some are really close to each other, and some parts of that basal ganglia are a little farther apart, but they're connected through axons, and they communicate with each other. Okay, those are called the basal ganglia, basal nuclei. Just mention about those just a little more specifically. The basal ganglia is this group of nuclei associated with many functions. Most of the time you'll hear to it as referred to as part of the motor circuitry. And so if people have Parkinson's disease, that's a basal ganglia problem. Part of that circuitry, when you send a signal to tell your muscle what to do, it doesn't go straight from your brain right to the muscle and through the spinal cord. It actually has to make a little loop through the basal ganglia first. And if that part of the basal ganglia that Parkinson's disease affects is called the substantia nigra. If you don't have enough dopamine that it produces, so the neurotransmitter to keep that signal flowing through there, it gets hung up. and you can't send the signal eventually down to the muscle, so it slows it down. That's why people with Parkinson's move a little slower, initiate movement slower, or it takes them longer to initiate movement because it can't get through this little basal ganglia loop. And you actually have the basal ganglia actually is important for cognition, for some memory, for vision. You have these loops, these cortical loops. So when your cortex has to process information, often sends it through the basal ganglia in a little loop. So anytime you have basal ganglia problems, you can have lots of other problems than just motor problems. But in the physical therapy world, we just talk about the motor part of that basal ganglia. But I want you to know that it has other roles. And then diencephalon, we already mentioned. It's mainly the thalamus and hypothalamus. Okay? Some books will include all the alamuses. There's a couple of other alamuses in your brain. There's the epithalamus and the subthalamus. Okay? However, the subthalamus is actually part of the basal ganglia circuitry. So really, diencephalon is just the thalamus and hypothalamus, but I just go ahead and I threw out those other alamuses there so you don't get them all confused. Those are kind of separate structures. They're important structures, but they're not part of the diencephalon per se. Okay? So the thalamus is like kind of the secretary of your brain. If you want to go up and communicate with the cerebrum, you typically have to go through the thalamus first. Okay? Even the basal ganglia, when it gets information looping through it, it has to go through the thalamus to get back up to the cortex. Or when sensor information comes in from my fingers or my toes, it has to go stop by the thalamus before it actually goes to my brain to make conscious awareness of it. So the thalamus is a relay structure primarily. It relays information, or it says, no, don't go up there. You're not important enough to get up to conscious awareness. So it's going to kind of be a little bit gatekeeper. Okay? Then the hypothalamus is very important for eating, reproduction, homeostasis, autonomic function. The hypothalamus is your main controller for autonomic regulation of just basic, feeding, reproduction, homeostasis, that kind of stuff, the hypothalamus. And also it's intimately connected with your pituitary gland. So here's deep inside the brain. You're kind of looking deep inside the brain. Actually, I just played the other day. We're going to get virtual reality, so we can I was able to play and go in. You can actually step inside. You've got a body in front of you, and you can have these little controllers. You can manipulate it, take it apart. You can actually stick your head inside somebody's brain and look around inside of this. It was pretty cool, and it's not that expensive to get some of the stuff. So we're actually going to do some research with that next fall with anatomy and neuroanatomy. So it'll make this stuff come a little bit more alive. So this yellowish structures here, those are the thalamus. You have one on each side. Most structures we have two of them or pairs of them. So you have a pair, a thalamus and a thalamus, and they're actually really close together. There's a little tiny space between the thalamuses that's actually a space filled with cerebral spinal fluid called the third ventricle, okay? But they're just really close to each other, and there's actually a little tiny stalk that sometimes connects them together, okay, called the thalamic adhesion. But not everybody has that, so must not be that important. And then just a little farther out, on the outside of the thalamus, are the basal ganglia structures. So the things in purple are the biggest parts of the basal ganglia. There's a couple other parts we're not showing there. The red thing on the end is the amygdala. It's not part of the basal ganglia. It has to do with fear, okay, and how we interpret and express fear is the amygdala. But the purple stuff here is the basal ganglia, okay? All I was going to show you here is just the diencephalon, thalamus and hypothalamus here, okay? You can't see basal ganglia here because it's farther, it's deeper, okay? You can't see it, it's deeper into the brain. It's actually it'd be inside here, but you'd have to cut away some brain tissue to get to that, okay? Your lobes, frontal lobe, everything in blue here. Motor and personality. It's just good to know this basic kind of facts, because then when you get questions about it, if you know motor and personality are there, then questions kind of you can make better guesses, educated guesses on things. I was good at that. I don't know. That's how I got through PT school. Educated guesses. That's how I got through most of my career. It's how I'm probably getting through today too. Temporal lobe, right here, lighter orange here. Memory and auditory information and processing. Okay, boiling it down to the big basic functions. Parietal lobe in green. Somatosensation, so feeling conscious awareness of your body, so being able to feel things consciously, and associating all kinds of different sensory functions and motor functions. You'll notice here's the occipital lobe. Notice how the parietal lobe has borders with the frontal, temporal, and occipital lobe. So it's good at bringing in auditory, visual, and somatosensory information with motor information and being able to make sense of it, okay? And knowing how to shape movement and how to process and say, because I have this sensation coming from my feet, it means I need to change the way my muscles contracting to keep my balance. So the parietal areas are really good at for this what we call association of integration and association of information. And then back here is the occipital lobe. It's big and it's primarily dedicated to vision because we are visual animals. I mean, we default, that's our primary sense. It's the quickest. We use vision very much, and it's got a big region devoted to it, okay? I told you about that insular lobe or limbic lobe. If you take away and you just either use your fingers or these little clamps and you pull away, and this is called the lateral sylvian fissure right here. If you kind of pull that open, you might be able to do it on some of these brains. Some of these brains are pretty beat up, so you may not. You can see some gyri and sulci a little deep in there. That's called the insular lobe. I think insular, it's insulated. It's kind of insulated by the rest of the lobes. You pull it apart, so this little insulated lobe, the insular lobe down in there, it's mainly autonomic and visceral control. So it's more of an autonomic structure. And maybe some limbic. So especially how emotions influence our autonomic nervous system. When you get flushed, when you're embarrassed, or you get butterflies when you're nervous, those kind of things. This is a fairly mid, you can see the cross section right here. You see how they show where they cut this through. This is a coronal section through the cerebrum. They've exaggerated this gray matter. See this gray matter right here? They made it nice and thick. It's not nearly that thick in real life, okay? And then all this stuff down here, this is axons, okay? So you got lots of little cell bodies up here. You also have some dendrites that are interacting with each other, but then all this is axons that are coming either up or down through the cerebrum, okay? This big structure here going back and forth, that's the corpus callosum that communicates from side to side. So corpus callosum there. These structures here, that's your thalamus on each side. This is your third ventricle, this space right here that has some fluid in it. These are your lateral ventricles here that have some fluid in them as well. These structures out here, a little more lateral, these are your basal ganglia structures out here. This is actually a part of the basal ganglia too. These guys are connected with each other. So this structures out here is basal ganglia structures, and this is the thalamus. Okay. This is part of the basal ganglia structure here too. This is your midbrain. This is the substantia nigra down here, but don't worry too much about that. Just giving you, just give you some orientation of this stuff. So that was cerebrum. Real gross overview of the cerebrum. We're going to do all the surface anatomy of the cerebrum at the end with the actual specimens. Okay. Now we're going to do brainstem. We're moving our way down. So we're in the brainstem. Get through that. We've cut away the brainstem here. So we're looking at this structure right here, and we're looking at the front of it. Okay. So we've cut away the big cerebral hemispheres up here. Okay. So what we have here is we have the midbrain, which is all this structure right here. Then this is the pons. And we're looking at the very front of the pons, and this is called the basilar pons. The bulbous, rounded front part's called the basilar pons. There's a little subtle depression here called the basilar groove. Your basilar artery actually lies right there, the basilar artery. And then down here is the medulla. Full name, some people call it the medulla oblongata, but most people just call it the medulla. Okay. Now let's get into a few structures here. We've torn all these off, but cranial nerves. There's a number of cranial nerves that are exiting from the front of the brain stem. I'll show you a picture later. We'll do a very couple slides on cranial nerves where you'll see where those exit. Okay, so Cranial nerves exit, at least 3 through 12 exit from the brainstem, okay? 1 and 2 exit from the cerebrum, okay? The optic and oculomotor nerve, the optic nerve and the olfactory nerve exit directly from the cerebrum. But from the brainstem, you've got cranial nerves 3 through 7, which have been basically all ripped off, so this looks like a nice, clean, easy to see brainstem. These structures right here, those are the cerebral peduncles. Remember we said peduncles are like connecting, and there's connecting stalks. They're a connection. Well, they are connecting the cerebrum to the brainstem. Cerebral peduncles. They're the connecting part or connecting area of the cerebrum to the brainstem. There's actually white matter tracts, actually mainly motor tracts running down through this area right here. Okay, that come down the front of the brainstem is some of your motor pathways in the cerebral peduncles. Yeah.
What's the difference between a cerebral peduncle and the midbrain?
So the midbrain is everything together, and including not just the front surface of it, so the whole thing. But that's a great question. So if we look down from the top of the brainstem, in a coronal section, we could have the midbrain, and we could also still see the cerebral peduncles, but they'd be just to the front of it. So only the front part of that midbrain is the cerebral peduncles. So that's a great orientation kind of question. In between here is this hole called the interpeduncular fossa. Okay, the interpeduncular. It's between the peduncles. It's a hole between the peduncles, so interpeduncular fossa. Okay, it's an opening or hole. These are not that important to us, but everybody asks what they are. Those are called the mammillary bodies, and if you look at the way I underline those, there's a reason why they kind of look like mammillary bodies. So this is kind of, I guess, the cleavage of the midbrain or something like that, if you want to call it that. So it plays a more role, some autonomic function and those kind of things, but it doesn't have a lot of other functional. Thank you, appreciate it. You see there's alternative terms, so the cerebral peduncles can also be called the crus cerebri or the basis pedunculi, depending on who you read. So interpeduncular fossa, we talked about the basilar pons, just this front part of the pons, this front surface of it, and the basilar groove is this little depression right here that the basilar artery is lying in right here. Down in the medulla here, we have the olives, which are these guys right out here. I guess you can imagine they look like olives somehow. Those bumps that sit right out there. And then we have what's called the pyramids. These structures right here that are kind of triangular or pyramidal in nature here, these are the pyramids. Okay. And what happens is your motor tracts that you guys have started to learn about, I know you guys started talking about some of the motor tracts, that come down from your cerebrum, come down through the cerebral peduncles, through the basilar pons, through the pyramids in the front of the medulla, then they get down here and they cross over to the other side. So this is where they're crossing, and there's a point right here where you have crossing called the decussation of the pyramids. Okay? And if you have a brainstem that you can take a probe or touch, if you take your probe and stick it in this ventromedian fissure right here, and you start running your probe right down the middle of it, you'll eventually hit a stopping point where these fibers start crossing, and you run into that, and that's the decussation of the pyramids. That's where those motor fibers start crossing, and that's why the left side of your brain controls the right side of your body from a motor standpoint, is because of that cross, where most of your motor fibers cross there, but 85% of them cross there. Okay, some stay on the same side, but most of them cross over there. That's the decussation of the pyramids. Backside's a little scarier, but we'll get through it. This is the backside of the brainstem. The first thing you're going to see here, don't worry about these guys up here. This is actually part of the basal ganglia sticking off here, is these four structures right here, which are the superior colliculi, these two structures here, the superior colliculus, and then the inferior colliculus. Okay? Those are little relay centers for reflexive control of vision and hearing. So like when a sound comes in and you hear a sound and your eyes reflexively dart over to look where that sound is, you know, it's almost like you have to, like, you know, when somebody walks in the back of the room, you know it's just somebody walking in the back of the room, but you'd have this urge to, like, divert your eyes to make sure it's not something dangerous or something you need to pay attention to. Those little guys play a role in that. So in that, even before that sound makes it to your conscious awareness, it goes, so the inferior colliculus is for your hearing, and the superior colliculus is for vision. So when your hearing comes in, it goes to the inferior colliculus and then connects with the superior colliculus, which then goes to a reflex center in your midbrain to tell your eye muscles, look that way, towards the sound, okay? So that's how that kind of works. So these together as a whole group are called the corpora quadragemina. Corpora, bodies, quadra, four, it's four bodies. The superior colliculus, there's two superior, two inferior. Together, they're the corpora quadragemina. So again, if you're reading and you hear that term, oh, they're just talking about the dumb superior and inferior colliculi all together, the corpora quadragemina. Let's see here. I'm going to point out this one cranial nerve because it's a little bit of a unique guy. See this little thing coming off the back here? That's the only cranial nerve that exits out of the back of the brainstem. It's called the trochlear nerve. Okay, it innervates one little muscle called the superior oblique muscle for moving your eye in a certain direction, down and in. But it's just kind of unique because it's the only cranial nerve that comes off the back of the brainstem, the trochlear nerve. Rhomboid fossa, this thing right here that I'm kind of tracing here, it's rhomboidal in shape here. That's a little depression or fossa, okay, in the back part of your brainstem. And what we've done, your cerebellum is normally sitting off the back here. The cerebellum's been cut and pulled off. And then deep to that is this rhomboid fossa. There's a space in there. There's actually some spinal fluid between your cerebellum and the back of your brainstem, and it fills that rhomboid fossa, okay, that little space there. So that's what that is. And remember how we had cerebral peduncles? That's how our midbrain connected to our cerebrum. On the back side here, we have some things called cerebellar peduncles, not cerebral, cerebellar. These are the connecting stalks to the cerebellum. And there's actually 3 different ones on each side, depending on how you cut the cerebellum off the back. This is how the cerebellum's connected. There's a inferior, middle, and superior. So middle is the big one. This is inferior, and this is superior. Cerebellar peduncles. So just the that's how your cerebellum's connected there, okay? Information that comes from your body to go to your cerebellum, like information from your joints and your muscles that tell you what your joints and muscles are doing unconsciously, it comes up through the back part of your brainstem and comes into that inferior cerebellar peduncle to get into the cerebellum. Once that information is processed, it goes out through the superior cerebellar peduncle to go up to the brain to tell you to make a correction in your movement if something's amiss. Okay, so don't have to know that. I'm just showing you why there's 3 of those, because there's entrance and exit from the cerebellum. The information comes in, information goes out of the cerebellum through different doors, okay, which are just these cerebellar peduncles. The other thing, last thing on the back side here, is the gracile and cuneate fasciculi and tubercles. Remember, a fasciculus or fasciculi, multiple fasciculuses, are where just fibers or axons are running. And tubercles are these bumps on the surface of the of a nervous system structure, and generally there's some cell bodies below these tubercles. Okay, you kind of have to imagine this a little bit, but there's a little bump right here. That's a tubercle. There's another little bump right there. That's a tubercle. There's a one right there, and there's one right there. And if you have a real specimen, you can kind of feel it, and you can kind of see where these bumps are. Okay, there's 4 bumps back there. The 2 that are most medial here, those are the gracile tubercles. The ones that are more lateral are the cuneate tubercles. There's little cell bodies below those. Okay, there's a little group of cell bodies. There's a nuclei there, group of cell bodies. Can you guess what the one below the gracile tubercle is called? What's this? The blank nuclei. There's the gracile tubercle, and so below it's called the gracile nucleus. Okay, so the gracile nucleus is just deep. When you look at it in a horizontal section, you see that little nucleus just below that. Okay, we're looking at a surface structure right now. The tubercle is the surface structure, but just deep in there is the nucleus. And then the same thing with the cuneate. There's a cuneate nucleus just below that cuneate tubercle. Okay, and then here is your fasciculus. I'm going to trace one out here. This little pathway right here, so running up from, this is a sensory pathway, running up the back part of your brainstem is called the fasciculus gracilis. Okay? That's carrying somatosensory information from your lower body. The gracilis muscles down in your lower body, that's how I remember, that's bringing up sensory information from the lower body. So the fasciculus gracilis is this more medial structure here. So the fasciculus is just below, there's axons. And they're coming up to actually then make a connection or synapse with the nucleus right there. You won't, we're looking like an X-ray or, MRI to deep in there to see what's going on in there, but we're just looking at the surface right now. Then this one just more laterally here, this is called the fasciculus cuneatus. Same thing. But that's bringing in sensory information from the upper body, okay? It gets layered on to the outside because you've got information coming from your lower body, up the back part of the brainstem there, the fasciculus gracilis, and then the upper body sensory information axons gets layered in because it's coming in a little later and gets layered onto the outside, and that's somatosensation from the upper body coming up the fasciculus gracilis to the fasciculus cuneatus, the nucleus cuneatus, and then at this level, all this information crosses over. This is where your proprioceptive information all crosses over in the brainstem. That's why we have our motor. We just talked about motor. The left side of our brain controls the right side of our body, but the left side of our brain is what's feeling what's coming up from the right side of our body. So it's the opposite. That's where at some point the information has to cross over to get to the other side. In this case, it happens right at that level for proprioception, not pain and temperature. This is just proprioceptive information from muscles and joints and so forth. Okay, so this is like the stuff that's telling you about where's my body. If I closed my eyes and said, my limbs are bent about this far, or I could match them up, or I know my position I'm standing, that's the conscious proprioception information that's coming up through that part of the brainstem. Pain and temperature is different. That's not what we're talking about here. It's in a separate area. Crosses over down in the spinal cord. This one crosses over up in the brainstem. Let's finish up cerebellum here and spinal cord, then we'll take a break. Cerebellum. Here's our cerebellum sitting off the back here, this big guy. Look how many little folds and folia and sulci this thing has. Do you guys know what the purpose of all those folds on sulci and on our brain or the cerebellum, what the main purpose of that is? What does that do for us? Increases surface area. We can put more neurons and cell bodies for more surface area to have more interaction between those. So this thing does a lot. When you got that thing is so enfolded and has so many little folia. And what it does is it processes cerebellum information coming up from our body that we don't have conscious awareness of. So the cerebellum is not a conscious structure. All that information, you do send a copy of that to your brain. We just talked about it when we talked about the fasciculus gracilis and cuneatus. That's getting the conscious level of awareness, knowing where your body's in space. Your cerebellum's also getting a copy of all those messages that don't make it to your conscious awareness. And it actually detects what's going on in your body, but the cerebellum also, every signal you send down from your brain to your motor system, you send another copy of that to your cerebellum. So what happens is when you go to move, for example, let's say I'm going to lift up this water bottle. If I didn't know how much water is in it, I'm going, if that's full, I'm going to guess that I've done something like this before. I have a motor memory. I'm going to send a signal of a certain amplitude to these muscles in my bicep and my finger flexors to go ahead and lift this up, expecting this to be full, okay? So I go to do it, and let's say I go to lift it up and it's totally empty. And if I were going to do that, if it was totally empty, I've already preset my muscles as if it was full. I'm gonna bring this to my face too fast. I might whack myself in the head with it. You know, if it's totally empty, because I was expecting I'd had them turned on too much. So what happens is when I did that, when I sent the signal to these muscles to preset them at a certain amplitude and force, I also sent a copy of that message to the cerebellum. Okay, so right at the same moment, right as I go to lift that up, all of a sudden it's too light, but my muscles are turned on too much right now. So all of a sudden, what happens is my bicep contracts too strongly, and the amount of flexion at my elbow is happening at an angular velocity that is higher than I expected it to. And so that signal, that sensory signal from the joint receptor and from my muscle spindles goes back to my cerebellum, and it compares the signal I just sent, the expected outcome, with what's actually happening. And it says, oh crap, you got to turn that down because you're going to whack yourself. So the cerebellum sends a signal back up to the cortex before I even have conscious awareness of what's happening to turn down the next signal to my muscle to dampen it. It's not perfect, because I might go, you know, you've done it where you go to whoa and you go to pick something up that's lighter. It's not perfect, but you don't fall over, you don't whack yourself with it. But there's a slight delay, but it's not too bad. So the cerebellum's like your anti-lock brakes in your car. It won't let you step on the brakes so you cause it to skid too much. You might still, you know, not perfect, but okay. It's got an anterior lobe is this area right here, and this big posterior lobe right here, okay, anterior and posterior lobes. When you look at it from the standpoint of, that's if you're looking at it in a sagittal section. When you look at it, here's a cerebellum that's been cut off the back of the brainstem. Here's that middle cerebellar peduncle, inferior and superior cerebellar peduncles. Same here. Okay. When you look at it in this view or from the top, this is looking at it from the top, the other picture, you can divide it into lateral zones. The middle part is called the vermis, and that means worm-like, and so it's this little worm-like area here. This is also called the vermis right here, so the very middle part is the vermis. And then right out here, you kind of have what's called the paravermal region. And then out here is the lateral hemispheres. Those regions like that in this kind of medial to lateral arrangement is kind of like the middle of your body to the more outside of your body. So the lateral hemispheres more have to do with your helping you coordinate and control your distal limbs and your hands and feet and your lower legs and distal arms. As you get more in the middle, it's more like your shoulders and your hips. And then as you get into the real middle, it's like your face and your trunk, okay? And that's what it helps to coordinate the muscles in each of those regions, okay? So it's kind of like a body, almost if you just kind of drew a person in here with their arms and legs kind of out like this. That's a little bit of a gross simplification, but that's kind of how it's somatotopically arranged, that the certain parts of your body are associated with certain regions of your cerebellum, the information they receive from those joints, those muscles, and those kind of things. All right, let's get through spinal cord. We'll take a break. You guys hanging in there? Spinal cord. Now we're finally out of we did the cerebrum, brainstem, cerebellum. Now we're lastly in the spinal cord, okay? Central nervous system. Spinal cord lies within the vertebral column in that canal. It extends to the level of the L1, L2 intervertebral disc space. So the spinal cord itself doesn't go all the way down. It stops partway down. So that's important from an injury standpoint, from diagnostic. That's the reason why when they do lumbar punctures, they put them in below that level so they don't hit the spinal cord. There's a nice space in there you can do, like take cerebrospinal fluid out or give somebody an epidural injection and those kind of things. You're not as going to damage or run into the spinal cord. There's an area of the spinal cord at the very tip where the nerves all come out at the end there to go to the lower part of the body called the cauda equina, horse's tail. It looks like a little bit of a horse's tail. There's a little tiny piece of what's called pia mater forms this thing called the filum terminale. Right off the tip of your spinal cord, it goes all the way down to your sacrum and anchors your spinal cord to the sacrum. This little, almost microscopic. it kind of strand that holds that, anchors it a little bit, called the filum terminale. We have, coming into our spinal cord, we have these different roots, okay, and going out as well. So the front part of your spinal cord, well, no, sorry, the back part is called the dorsal roots, okay? So when information from your peripheral nerves coming in about sensory information goes in the back door, okay? So it goes through the dorsal roots. Afferent sensory information goes into the back part of the spinal cord through the dorsal roots. Motor information and autonomic control, okay, goes out the front, the anterior roots, okay? Then those end up forming these spinal nerves. The roots come together very quickly to form the spinal nerves, okay? And we have 31 pairs of those, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. So main things here, here's all of our nerve roots coming out. They're different respective levels. There is the end or tip of the spinal cord right at that L1, L2 disc interspace right here. The disc is right in here. Actually, no, the disc is in front. That's the back. So the disc is right in here, L1, L2. Right in that area there is where you see that end. And then all this stuff coming off are the nerve roots that have to still go down all the way to their exit points from the vertebrae at the different levels. They still have to extend down there, and that's the cauda equina. That's all those nerve roots that are hanging off the bottom there. They still have to go down and descend and exit out at their appropriate level from the spinal column. Okay, out the little holes, the little foramina or little holes they exit out from on each side. This is just a frontal view with the spinal canal and column removed here. And you can see how the cervical area of the spinal cord is a little bigger. It's called the cervical enlargement. And the lumbar area is a little bit bigger because there's just more stuff coming in and out. You think about your cervical area, that's all your hand and arm function. So you have a lot of sensory information coming in, plus you have a lot of control of a lot of little muscles and so forth. And so there's more stuff that has to happen there. So it's bigger. There's more gray matter there to process that information initially. Same thing with the lumbar area. Okay, the thorax area doesn't have nearly as much stuff as far as muscle control and sensory information coming in, so it's a little smaller and narrower. So now we're going to look at a cross section, that horizontal section through the spinal cord. And remember how in the cerebrum, the gray matter is on the outside with the white matter on the inside mostly. This is the opposite. Gray matter, cell bodies are mostly on the inside and all the white matter is on the outside. So the gray matter is this stuff. This H-like structure here is the gray matter. Okay? You have a dorsal and ventral horn. So this is the dorsal horn back here, and then the ventral horns are here. So in the dorsal horns, information comes in through the dorsal roots and often makes a first synapse here, okay? Especially if it's a pain and temperature. It's going to make a synapse here. Sometimes it comes into that dorsal horn and goes up the back before it makes a synapse, especially if it's a proprioceptive type fiber. So sensory information comes in through the dorsal horn and dorsal roots. Motor information, you have cell bodies here that are going to go out the front part of the spinal cord. Those are your anterior horn cells because they're in the anterior horn. They're your motor nerves. They're going to go to the muscles in your body and come out the front part of your spinal cord, okay, in the ventral roots. So then that's the gray matter. Then the white matter is all your axons. Okay, this is your ascending and descending track. So once information comes in, it's going to go up, or sometimes it's coming down through the spinal cord, or coming down here, and then communicating with anterior horns here, if this is the lateral cortical spinal tract. So there's information going both ways, passing each other. It's amazing how it's all layered. I think I have a slide in here at some point that shows how it's all actually layered in there. This is just helpful to look at as how the spinal cord is sitting within the vertebral column. Okay, so we're looking at a horizontal section at a certain level. This would actually be a thoracic level because I can just tell by the gray matter, the shape of the gray matter here. So here's the spinal cord. You have some coverings of the spinal cord. There's little purple coverings here. That's the meninges. So this is the pia, arachnoid, and dura, but don't worry about that now. Just know I'm just pointing out that those are just the protective layers that we have also around our brain, but they're around our spinal cord. This is that dorsal roots. Okay, you have these little roots that come out at each level. So back here's the dorsal roots. Then here's the ventral roots out here in front. Fairly quickly, once you get through the little lateral foramina here where it exits out from its vertebral level, those come together into what's called the spinal nerve. Okay, the roots come together. And once those roots come together, these nerves that come out are mixed nerves. They have sensory, motor, autonomic, if they're coming from the thoracolumbar area, information inside of those nerves. Back here in the dorsal root, it's all sensory. In the ventral, it's motor. Okay, but then they join and they split into a dorsal ramus and a ventral ramus. The dorsal ramus is your motor and sensory going to the back part of your body, and the ventral ramus is the motor and sensory and autonomic going to the front part of your body. So very quickly that spinal nerve kind of divides into two major groups. One goes towards the back of the body, one goes towards the front of the body. Don't worry too much about these little guys. These actually are going to your, this is related to some sympathetic nervous system function. If you're in this thoracic level, but those are some little ramuses that come off and your sympathetic fibers have to go through there, but don't worry too much about that at this point. Let's take a break. I'm about done for the next few minutes here, and you guys are about done. Any questions before we take a break? You guys hanging? Sorta? All right. Come back and let's give you 10 minutes. Good 10 minutes. We'll start up again at 2:30. A whole 10 minutes. Wow. All right. Thanks for coming back. I was afraid some of you might not want to come back. I didn't want to. I had to work myself up. All right. So, you guys doing okay? Any questions you thought of while you're wandering around? We are going to keep going, and so we've talked about cerebrum, brainstem, cerebellum, spinal cord, kind of gotten through that. Now we're going to talk about a little bit about the coverings of the nervous system. I've already mentioned them a few times in passing. We're going to talk about the coverings, and then a little bit about blood supply, and then a few clinical things, and then we'll get the brains out and start doing surface anatomy. So, coverings. Collectively, these things that cover our central nervous system. We're talking about the central nervous system. So these coverings cover our brain, brainstem, cerebellum, and spinal cord. These are surrounding all those structures. They're collectively called the meninges. Okay? So when you hear somebody has meningitis, that's an inflammation or infection of the meninges or the dura and arachnoid, so the meninges. You have 3 layers. Okay, if you go from the innermost layer to the outermost layer, so the innermost layer is the pia, so actually the last one on there, they form what's called a pad. So PAD. Pia is the innermost layer, A for arachnoid, then D for dura mater. Okay, so from inner to outer, that's kind of how I remember them. They're a pad. Pia, arachnoid, and dura. But starting with the outermost layer right now, the dura mater, which literally means tough mother. And it's tough, and it's like beef jerky, sinewy, leathery, almost feeling kind of structure. Okay, the dura mater, it's the outermost membrane. It forms a couple important structures, like the dura goes down in between your cerebral hemispheres, then the cerebral longitudinal fissure, and forms this structure called the falx cerebri. And it's made out of dura mater, but it's called the falx cerebri, is that dura mater going down in between just until you get to your corpus callosum. So it's called the falx cerebri. Part of your dura mater goes up in between the cerebellum and your cerebrum. So your cerebellum's sitting there below your cerebrum, and there's some dura that slides in between there, and that's called the tentorium cerebelli. It looks like a tent over the top of the cerebellum, the tentorium cerebelli. And if you guys get into different, like when you're doing a neurorotation or whatever, and they talk about a supratentorial lesion is like a stroke or a brain injury or trauma to the brain above the tentorium cerebelli. An infratentorial lesion is something like damage to the brainstem or the cerebellum. So supratentorial is mainly to the cerebrum. Infratentorial lesions below the tentorium cerebelli are brainstem and cerebellar lesions typically, okay, or problems. The dura mater also forms these drainage ditches, basically. The layers of your dura mater form these things called sinuses, the dural sinuses. And so your venous drainage, all the blood from your brain, once it's been used, it starts in some veins in your brain or on the surface, but all that eventually dumps into these dural sinuses. Those veins dump into these drainage ditches or sinuses that are formed by the dura, these tubes of dura, okay, that finally gets down into your body and the jugular vein and so forth, okay? The arachnoid mater, arachnoid, spider-like, it does kind of look a little spider-like and web-like. Some of these brains still has some arachnoid on them. So you take the dura off, and then you have the arachnoid sitting there. And when you have normal cerebral spinal fluid, there's the cerebral spinal fluid. It goes from inside of your brain and gets to the outside of your brain and spinal cord, and it flows in a little tiny space between the arachnoid and the pia, okay? There's a little space. When you have cerebral spinal fluid in there, there's a space. When you don't have cerebral spinal fluid, that space goes away. You have to actually have cerebral spinal fluid in there to have that space there, okay? So that's the arachnoid, and the subarachnoid space is between the arachnoid and the pia. The pia is a microscopic layer. You can't see the pia on the surface of the brain, cerebellum, brainstem. The only place you see the pia is where it forms what's called the filum terminale. It comes off the tip of the spinal cord to anchor to the sacrum, and then on the edges of the spinal cord, some pia extends off the edges of the spinal cord at different points to anchor it to the inside the spinal canal at each level, and that they're called the denticulate ligaments. They're pia mater, those little extensions of pia off the edge of the spinal cord to anchor it to different locations in the spinal canal. Come on, go forward. There we go. All right, so this is like a little cross section of the top of a brain here, and so here's the skull or your bone right here, this and skin, most outer layer there. But then here's the dura. The pink is the dura. Okay, just below the dura is the arachnoid, and there's these little end feet that come down from the arachnoid, and it's kind of a clear, fibrous-looking structure. It looks like a spider web a little bit, but then inside here, this is where the cerebral spinal fluid is running in that little tiny millimeter or two thick space, the subarachnoid space, and then that layer that's right on top of the cortex there is the pia, okay? Then you can see here is your blood vessels that run on the surface of your brain also run in that subarachnoid space. So when somebody talks about somebody having a subarachnoid bleed, that means the artery, one of the arteries on the surface of your brain between the arachnoid and the pia is bleeding and bleeding into that subarachnoid space. And eventually that blood will get into the cerebral spinal fluid, and then actually when they do a spinal tap and they pull, they're looking for blood cells to identify if they truly have a subarachnoid bleed, because it actually goes and flows, your cerebral spinal fluid flows around your entire spinal cord, so they can go down to that below L1, L2 and take some cerebral spinal fluid and see if there's any blood cells in there. That's one way. Of course, MRI will also show subarachnoid bleeds. So here's the back of a skull or back of somebody's head, and you're looking at the back of the brain. This is, you can see this is skull right here. They've actually cut away. This is the foramen magnum right down here. This is the foramen magnum. So we've taken off most of the dura. So the dura was normally right here. We've ripped or cut most of it away, but we can still see the edges of the dura that comes down in between the cerebrum, called the falx cerebri. Okay, so that dura that dives in deep in between the two cerebral hemispheres is called the falx cerebri. And then this white stuff here, this is the dura that dives in between the bottom of the cerebrum and the top of the cerebellum to form this thing called the tentorium cerebelli. So it's kind of like a little bit of a, not doing a very good job there. tentorium cerebelli. So lesions up here in the cerebrum are supratentorial lesions. Lesions down here in the brainstem and the cerebellum below that is this infratentorial lesion. Okay. Here is looking at a sagittal section, a mid-sagittal section down through the middle of the brain, and what they've done here is left the falx cerebri. So this is the dura that extends down. Here's the corpus callosum right here. It extends down to the corpus callosum, okay? And this is the tentorium cerebelli right here that goes over the top of the cerebellum. So this whole structure here is the falx cerebri. This is the tentorium cerebelli. And then you can see the dura extends all the way around the whole spinal cord and brainstem, and cerebrospinal fluid runs in that subarachnoid space that's in there. Okay? Blood supply. Real quickly on blood supply. We got to have blood going to the brain. That's got to be a constant flow. And you know, one interesting thing about the brain is we don't store glucose in our brains, so you have to have constant blood supply for your brain to function. That's why, you know, so you stop blood supply to the brain for just even a few seconds, and that's why you pass out, you know, is because you don't have any glucose to run your neurons off of, only what's available at the moment from your blood supply. Okay. So the internal carotid arteries are your big ones, okay? They provide blood to most of the cerebrum. Okay, the big part of your cerebrum is the internal carotids. As they come up into your cerebrum, they divide into the middle and anterior cerebral arteries. Okay, that supply most of your front and outside of your cerebrum. We also have the vertebral arteries. So the vertebral arteries are coming up on each side of your neck and each side of the spinal column, okay, through the vertebral foramen. They come up and then join together to form the basilar artery, and eventually they terminate or end as the posterior cerebral arteries. So we'll show you a picture here, I think, that makes more sense. Pictures are always better. As I mentioned before, eventually that arterial blood has to get out of your brain once it's been used to the venous system. So there are veins deep in your brain and veins on the outside of your brain, but all that venous blood eventually dumps into one of the sinuses, okay, the dural sinuses, which form these tubes that gather this venous blood and eventually drain it into your big jugular veins. This is the simplest picture I can show you here. So here's the internal carotid artery coming up from in your neck, and it comes up and divides into a middle cerebral artery that supplies most of the outside surface of your cerebrum, and an anterior cerebral artery which supplies most of the medial surface of the cerebrum. Okay, so this is kind of the dividing line between the circulation. This is the anterior circulation versus what we call more the posterior circulation. This is your vertebral artery. You have two of them, a pair of vertebral arteries coming up on each side of right next to your spinal column, okay? They join to form this basilar artery right in front of the pons there, that basilar groove. Remember I said that's where that basilar artery sits, this big basilar artery? Eventually it terminates into the posterior cerebral artery on each side. There's two posterior cerebral arteries. They go back to supply this back and underneath the surface of the cerebrum, a small percentage of the cerebrum. But all of this, the vertebral and basilar arteries, they supply the cerebellum and the brainstem and the upper part of the spinal cord. Okay? Actually, they supply most of the spinal cord, not just the upper part. Just point out a few structures here. So here's kind of looking at the front of somebody looking at you with just their vascular system here. So let's point out the major structures here. So these guys right here and here on each side here, those are your internal carotid arteries. Okay, that's the internal carotids coming up. That goes to your brain, the cerebrum, that anterior circulation of the cerebrum, the internal carotids. The external carotids go to the outside of your head and face. They're supplying the arterial supply to your head and face and muscles and stuff in the face. But the internal carotids are going up. So the internal carotids come up here, and they divide into, this is that middle cerebral artery right here that supplies more of the outside of the cerebrum, and this is the anterior cerebral artery that supplies more of that medial or inner side of the cerebrum, okay? So that's the internal carotid comes up here and divides it into the two branches of the middle cerebral artery and the anterior cerebral artery. So internal carotid, middle, and anterior cerebral artery is supplying most of the cerebrum, okay? Erase that. Here is your two vertebral arteries running up on right on each side of the vertebrae. In this little, there's little holes that actually these transverse processes. There's a little hole that those run up through, called the vertebral foramen. Okay, the vertebral artery runs through these openings called foramen and holes. This is where, like, you worry about somebody if they have a lot of arthritis and little spurs in there. Those can poke into that vertebral artery. So if somebody goes and gets a manipulation by a physical therapist or a chiropractor who doesn't know how to test for that or isn't cautious, you can tear or cause a vertebral artery dissection, where you rip that or pinch it enough that it causes a clot, and that's not good. It's a stroke. So the vertebral arteries join together once they come off the spinal column there into the basilar artery right here. Then the basilar artery goes and it dives backwards into the two posterior vertebral arteries. So these guys are kind of diving backwards to the bottom part of the cerebrum and the back part of the cerebrum. Okay. But as it's doing that, it's also giving off little collaterals here to the brainstem and cerebellum. Okay. So we've got the internal carotid system and the vertebro-basilar system. Okay. Those are your two main blood supplies. Now, there's a connection between the vertebro-basilar system, the internal carotid system, called the circle of Willis. You guys heard of the circle of Willis? Okay. This is an anastomotic ring. It's an anastomosis, or connection between multiple blood vessels that form this ring. And so there's a little connection right here between the two anterior cerebral arteries. That's called the anterior communicating branch. Then you have these two little posterior communicating branches that communicate between the internal carotid and the posterior cerebral arteries. So now we've got this contiguous connection between all those different arteries. So what that means is that we have potentially alternative routes for blood supply if one of those gets, bottled up. So for example, let's say, and this, I've had patients walk in the door that look totally normal, yet they have a 100% blockage of their internal carotid artery. And you're going, holy crap! You should be, you should be able to move your entire. If this is a left internal carotid, you shouldn't be able to move your right side of your body. You've pretty much destroyed the blood supply that goes to that entire part of your cerebrum. Why are you still walking? Why are you still alive? It's because this person was lucky. They had good anatomy, or that clot took a long time to develop. So like there's a certain type of clot called a thrombosis. So over time, this clot develops slowly over time, and if that clot is slowly narrowing that artery, your blood supply says, uh-oh, I need to start rerouting. I'm getting too much resistance here. I'm going to reroute. So blood supply from this side can make it around to the other side through this anastomotic ring to get to the other side. But that requires them developing some extra patency or dilating those vessels that allow, and some people's anatomy is good for that. Other people is not as good. They're not as lucky. Or if somebody has a big stroke right away, your system doesn't have time to adjust. And so if you have a big clot that all of a sudden forms, and you would have it would be much more difficult to reroute blood to that side and prevent long-term damage. Okay, but there are people walking around with 100% occlusion of a big internal carotid, and they're just lucky that their body adapted and rerouted blood supply around through that circle of Willis to supply the region that wasn't getting it. That's what this is the circle of Willis again. This is right here. These structures right here. This is the internal carotids coming up right here on each side. This is the posterior cerebral arteries that are ending off the basilar artery, and there's the communication between these is to the internal carotids, and the posterior communicating arteries is called the posterior communicating artery that is between the posterior cerebral and internal carotids. That's how you can get blood supply there. And then this guy up front here is the anterior communicating between the two anterior cerebral arteries, and that's how blood can flow around and get past clots and blockages. Thinking about bleeds and so forth as it relates to the different layers, especially the meninges, and this is kind of relates to the vascular system here. We have epidural hematomas. So here's an epidural hematoma. Here's the dura. See the dura is that little area right there. This is where you have a blood vessel that is damaged that is on the outside of the dura, like in your skull or just under the surface of the skull, between your skull and the dura, okay? The big one that tends to cause a problem here is the temporal artery. Right in your temporal bone, and there's an area of your temporal bone that's a little thinner, and if you get whacked right there by a ball or by something that hits you right on the side of your temple there, that temporal artery that is between the bone and the dura can bleed, okay? And it's a pretty big vessel, and it'll bleed fairly rapidly. Initially, the person thinks they're doing just fine, you know, hey, I'm doing okay. But it's an arterial bleed. Arterial bleeds are high pressure, so they can quickly go downhill. And so this is where you hear about, what was it, Liam Neeson's wife, Natasha Richardson. Did you hear about her a few years? Skiing accident, hit her head. She thought she was doing fine. Everybody thought she was doing fine. But within an hour or an hour and a half, she's dead because she had an epidural hematoma. And by the time you catch it, it's sometimes too late, because what they need to do is they need to drill a hole in there and just takes that blood out. And then you can be totally fine from that. It's usually not a problem as long as you recognize it and create a little burr hole and let that blood get out of there, okay, and drain that out. But it happens pretty quickly because it's arterial, high pressure, and it can just, eventually what happens, your brain gets pushed down into the foramen magnum and causes your brainstem to get squeezed down in there. In your brainstem, you have your respiratory and cardiac centers, and eventually you lose consciousness and you stop breathing because you get pressure of everything being pushed down because of the high pressure and space that this is taking up. Then we have what's called a subdural hematoma. So this is the dura is still out here attached to the skull here. This is a venous bleed. There's actually little veins. There's veins that come from the surface of your brain that are going out to this dural sinus. Remember I said there's the sinus right there? That's the dura forming that tube that your veins are going into from the surface of your brain. A subdural hematoma is most common in an older adult who oftentimes has some dementia too, because their brain is shrinking. And what happens as your brain shrinks, it's taking up less space. It's pulling away from the surface of your cranium there and the surface of your skull, and there gets to be some space in here. And what happens as your brain atrophies, this is somebody who's got a little bit of brain atrophy, if they get a slight little bump, and even sometimes it happens spontaneously, the little veins that were going from the surface of your brain into that dural sinus, they start to get stretched because the brain is being pulled away from that, and they start to tear. And little veins start to tear. And over time, that space gets filled up with venous blood. But the thing about this is it usually is not an emergent situation. Sometimes people have these for years, and they get this big dried clot right there, and only by chance do they find an MRI because it's a low-pressure system. It doesn't create this dramatic emergent need. It doesn't push the brain as hard down in the foramen magnum because it's just this slow little leaking of venous blood that just tends to clot on its own over time without creating a big problem. Now, it can eventually cause a problem, but they usually just look at them and say, yep, you got this subdural hematoma. We're going to watch it for a month or two and just make sure it doesn't change. As long as it's stable, there's nothing we really are going to go in and mess with that. Okay? That's a subdural hematoma. The other type of hemorrhage we can have is a subarachnoid hemorrhage, okay? Subarachnoid hemorrhage is where you have that hemorrhage of some of the arteries in that space between the arachnoid and the pia, okay? Those tend to be bad too. A subarachnoid hemorrhage is usually it's an arterial hemorrhage on the surface of the cerebrum between that arachnoid and pia layer. And that is emergent, and you need to do something about it, and that will cause a bleed that can lead to, again, damage in the brain and a stroke. The last type of hemorrhage we can have is called an intracerebral. So this has nothing to do with its relationship to the different layers or meninges. This is a bleed right into the deep part of the brain of a deep penetrating artery into what we call the parenchyma. All the white matter and neurons and glial cells in there, that we call that the parenchyma of the brain, the deep part. So that's just a deep cerebral bleed or intracerebral hemorrhage. Has nothing to do with a hemorrhage on the out. The other ones are on the outside of the brain and how they relate to the different meninges, where the bleed is happening. Outside of the dura, just below the dura, or below the arachnoid. Then we just have bleeds that happen inside the brain itself, okay? Most aneurysms, like if you have an aneurysm that bursts on you, a lot of those are subarachnoid. Those happen to be at bifurcation points, like where the internal carotid and the middle cerebral artery come off, and there tends to be in that subarachnoid space. Ventricles. We've seen them in a few of the cross sections. So that was the vascular system and a little bit of a clinical stuff on bleeds. Now the ventricles are those things that are these spaces inside your brain and inside your, even in your kind of brain stem area and even in your spinal cord, where you have little holes or little spaces that are filled with cerebral spinal fluid, okay? Eventually that cerebral spinal fluid does get to the outside into the subarachnoid space, and we'll show you kind of how that happens. The ventricle system, primary function is the regulation of the extracellular fluid and protection of the CNS. So it's kind of like your brain and spinal cord are suspended kind of in this neutrally buoyant little, tank of water or cerebral spinal fluid that kind of pads it and keeps it from getting knocked around too much and gives it that support, okay? So you make cerebral spinal fluid by filtering your blood. Okay, it's basically almost like a plasma. Okay, it's a clear liquid that is just filtered by your blood, and it's filtered by this structure called the choroid plexus. It's this little granulated-looking tissue that's little like capillaries that filter blood and make the cerebral spinal fluid that goes into the ventricles. Okay, you have 4 ventricles. Okay. You have the lateral ventricles, which are your ventricles essentially 1 and 2. You have 2 lateral ventricles. Then you have 1/3 ventricle in the middle and 1/4 ventricle, okay? And connecting the 3rd to the 4th ventricle, you have the cerebral aqueduct, and we'll show you a picture of this. We've already mentioned the dural sinuses. The cerebral spinal fluid, just like your venous blood drains into your dural sinuses, you recirculate your cerebral spinal fluid. Your cerebral spinal fluid also goes into those dural sinuses and is recirculated by your system, okay? So the things you see in blue there are the ventricles. So within your brain are these spaces. They're pretty big and pretty large spaces. And these are the horn-shaped curved things here on each side. These are the lateral ventricles, okay? Your basal ganglia and thalamus are sitting kind of just on the inner surface, just kind of below and inside of those structures, okay? Then this is right here is your 3rd ventricle, right between the 2 thalami. Is this little tiny sliver of a space, okay, is the 3rd ventricle. Then you have a little thing right here, a little connecting tube called the cerebral aqueduct, which is actually in your midbrain. Then once it gets down in your midbrain, it opens up into this space that's between the back part of your brainstem and your cerebellum. Remember that rhomboid fossa? That's the rhomboid fossa forms what we call the floor of the 4th ventricle. It's kind of like the floor of the 4th ventricle. Okay, and you can see this is actually, the 4th ventricle is rhomboid in shape. So that's looking at it from the front. Here's from the side right here. You can see here's our nice curved lateral ventricles, which have an anterior horn, an inferior horn, and a posterior horn to them. Here's that third ventricle right here, that little sliver of a space. This is where that interthalamic adhesion, so that's where your two thalami might be connected right there, so it kind of invades that space a little bit. Cerebral aqueduct is the little tube here, and then this is your fourth ventricle right here, the thing sitting right here between the back part of your brainstem and the cerebellum sitting off here. That space between them is the fourth ventricle. Okay. That stuff we called choroid plexus, the little capillaries where it filters your blood to make the cerebrospinal fluid, you tend to have a lot of it right here in the floor. It sits right here in this little granulated tissue in the floor of the lateral ventricles, and also in the fourth ventricle. You tend to have some in your fourth ventricle, some choroid plexus. So once that cerebrospinal fluid is formed, it migrates and it goes through from the lateral ventricles. It goes down into the third ventricle through these little openings called the intraventricular foramen. Then once it goes through the third ventricle, it goes down through the cerebral aqueduct, goes down into the fourth ventricle, and then there's three little holes below the bottom of your cerebellum. There's two on each side, and there's one right in the middle. The cerebrospinal fluid then goes from inside the brain and gets to the outside and starts to circulate all around your brain, spinal cord, brainstem, cerebellum in the subarachnoid space. But it gets out into that subarachnoid space through those three little holes just below the bottom of your cerebellum, two in the very outside and side, kind of inferior and lateral, and one right in the middle. Okay. Then eventually that cerebrospinal fluid gets into the dural sinuses and gets reused, okay, recirculated and recycled. Okay. So you're constantly making cerebrospinal fluid and putting more into your system, and it comes back out. Okay. Here's a nice thing showing kind of the pathway of how all that happens. So lateral ventricle, it's produced mostly, goes into the third ventricle, cerebral aqueduct into the fourth ventricle, then it gets out these little holes below the cerebellum into the subarachnoid space. It circulates all the way down the spinal cord too, but then eventually it circulates back up, and there's these little things right here called arachnoid villi that poke into the dural sinuses, and that's how that cerebrospinal fluid gets back into the dural sinuses and gets mixed with your venous blood and goes back into your system. Okay? If you have a blockage, this is the problem. You see how this little tiny tube right here, that's a cerebral aqueduct? That's a vulnerable place. If you're born without a fully formed tube, or you get enough pressure on a part of your brain that closes off that tube, all this cerebrospinal fluid starts to back up, and then these ventricles start to get bigger, and eventually you only have so much room. You got a skull, there's no more space. You start to push on brain tissue, and or if you're young and you haven't formed your sutures yet, you haven't fused the sutures, you get hydrocephalus. This is where the kids with the large skulls, because their skull can still accommodate it because you haven't fused yet. But if you're an adult, you're dead. If that starts to happen, you get too much pressure, you can start to have problems. Some older adults get a syndrome called normal pressure hydrocephalus, so they get some blockage or narrowing, and it's a very slow buildup of pressure in their cerebrospinal fluid system. It usually starts with incontinence. They get confused, their walking goes bad, and I've seen where patients took them a while to figure out what was wrong, but once they figured it out and drained that, all of a sudden they went from being almost non-functional to miracle, like I can walk again, by just draining off some of that cerebrospinal fluid. But it's called normal pressure hydrocephalus because it's not a real high pressure problem. It's kind of formed slowly over time. A lot of these people are also older adults that have some space where their brains are shrunk a little bit, so it takes a little while to build up enough pressure to start causing problems. Good? Okay. Now we're going to review a little bit what you guys talked about, some of your pathways. Did you guys start on pathways this morning a little bit? We'll just really quickly touch on those. We've mentioned them already a little bit. So when we talk about sensation from the body, we can talk about a couple main ways that sensation gets up to where it needs to go. Okay. The first being what's called the dorsal column medial lemniscal system. And the type of information this carries is called conscious proprioception. This is where you understand and know my body in space, or somebody touched me right there, or I moved my finger this much, or that kind of stuff. That's conscious awareness of proprioception. This is coming from joint receptors, muscles, tendons, ligaments, those kind of things that have special receptors that tell you where your body is in space and how fast your joints are moving and those kind of things. And you're consciously aware of it. It's getting up to your cerebrum. That's via the back part of your spinal cord in the dorsal columns. That's why it's dorsal columns. And it changes names, just like a lot of street names in Dayton, Ohio. You cross over one thing. Once you get into the brainstem, it's called the medial lemniscus. So that's why it's called the dorsal column medial lemniscus. It starts out in the dorsal columns at the back part of the spinal cord. Then it still goes up the back part of the brainstem, but once it's in the brainstem, it's called the medial lemniscus. Okay? That's conscious proprioception. Then we have the spinothalamic tracts, or tract, depending on what you read, / anterolateral system. Okay? Some books will just call it the spinothalamic system or spinothalamic tract. Some books will call it the anterolateral system. The spinothalamic tract is just one component of this whole anterolateral system. It's actually, you can kind of break that tract into some multiple parts, but let's just think of them as synonymous right now. Anterolateral, this pathway in the spinal cord runs more in the front and lateral, anterolateral part. Okay? The pain and temperature pathways go up the front part of the spinal cord. And then they eventually get into the brainstem. In the brainstem, they're actually called the spinal lemniscus, kind of like how the other one called the medial lemniscus, medial lemniscal system, dorsal column. You could call this one spinothalamic / anterolateral / spinal lemniscus if you wanted to. But remember, lemniscus as just a flat ribbon-like structure. It just means that once you get up into the brainstem, the axons tend to get a little more flattened. They're not as round. They kind of get a little more flattened in shape. The last big, so that's pain and temperature. That's conscious pain and temperature. You're aware of this, that somebody poked me right there and it hurts. Then the other pathways, there's a group of pathways. Collectively, we're just going to call them the spinocerebellar tracts. There's actually 4 separate ones, but we're just going to collectively call them the spinocerebellar tracts. And it makes sense. Spinocerebellar. Look at the names. It starts in your spinal cord, and it ends in your cerebellum. That's the unconscious proprioception. That's the stuff that you don't even know is going on. That's that background check system we talked about in that example I gave you. Spinocerebellar. If you look at spinothalamic, that makes sense. Spinal cord up to the thalamus. It's definitely going to go to the if you go to the thalamus, then you're going to go talk to the brain at that point. But sometimes the terms help you know where it starts and where it's going. If you hear something like spinocerebellar or spinothalamic, you're thinking it's starting in the spinal cord and going somewhere more proximal or central, so that's more of, they're afferent sensory type pathways, okay? Here we go. So here is your dorsal columns, medial lemniscal system right here. This is proprioception. Here's coming in from the leg, goes in the back part of the spinal cord, goes up the back dorsal columns here, gets into the brainstem. Once it's in the brainstem, this part right here is called the fasciculus gracilis. That little bundle or packet of axons, the fasciculus gracilis. Its synapses here in this little nucleus called the nucleus gracilis. Remember when we looked at the back part of that medulla, we were looking at the tubercle there, the gracile tubercle. Makes a connection or a synapse. Oops. Crosses over into this structure called the medial lemniscus. So this is the dorsal columns. The dorsal columns are the fasciculus gracilis and cuneatus together, are the dorsal columns. Crosses over in the medial lemniscus, goes up to the thalamus, because you got to talk to the thalamus before you go and finally talk to the cortex. Okay. This has got 3 neurons in this pathway. It's got a first order, second order, and third order neuron. Okay, so there's 3 neurons in this particular pathway. These cross, so it's opposite side. okay, and it crosses in the medulla, or in the brainstem region, is where the proprioception crosses. Here's the upper body from your hand. It comes in the cervical spine, goes up more laterally, but in the dorsal column, in the fasciculus cuneatus. Synapse is in the nucleus cuneatus, then crosses over, becomes one with the medial lemniscal system here. The medial lemniscus goes up to the thalamus, then up to the cortex. Okay? And actually, this is not a good example, because if it was coming from the leg sensation goes over here. That's okay for upper body, but this is not a good schematic, because your leg is actually right here. This should be a foot right there. It's on that side of your cerebrum. So don't ignore that where its final destination is. Here's pain and temperature. So they're showing you somebody getting burned or stuck with a needle or something like that. So when it comes in to the spinal cord, this is coming from the lower body into the lumbar spinal cord, it makes an initial synapse right away. It doesn't go up the dorsal columns like the proprioception did. It makes a synapse in the dorsal horn and then immediately crosses over. Crosses over right here, called the anterior white commissure. Crosses over, and then it goes into the spinal thalamic tract, which is part of the anterolateral system, anterior and lateral, and then it stays in that position kind of all the way up. Eventually in the brainstem it moves a little bit more back, but it also goes to the thalamus, because it has to go to the thalamus, because it's going to go up to the cortex. Legs should go over here. Upper body's over here. Okay? So but the crossing over happens in the spinal cord. It crosses over, but it happens in the spinal cord. So pain and temperature crosses in the spinal cord to the opposite side. Proprioception crosses in the brainstem, okay, in the medulla, particularly the lower medulla, but you don't have to know that detail. Lastly is the spinocerebellar tracts. This is a Golgi tendon organ, or it could be a muscle spindle. So this is like more in the tendons or muscle spindles. They go into the spinal cord. They go up the back part of the spinal cord and go into the cerebellum on the same side. They don't ever cross over. So that's the key with those. With the spinocerebellar path, they're not contralateral. These pathways are both what they're called contralateral pathways. One side of the body goes to the opposite side of the brain. These are ipsilateral, spinocerebellar pathways. The clinical significance of this is, if I have a patient with a stroke that involves right here, on the right side of their brain, they're going to have sensory problems on the left side of their body. They're going to have contralateral symptoms to the lesion. The lesion's on the right, the symptoms are on the left. Contralateral sensory loss. Same thing with pain and temperature. Contralateral loss if we've got a lesion up in the brain that's on the opposite side. However, in the cerebellum, if I have a stroke or a tumor in my right cerebellum, I'm going to have some incoordination more on the right side of my body. I'm going to have an ipsilateral loss of coordination because those pathways don't cross. So if it's a cerebral lesion, contralateral deficits. If it's a cerebellar lesion, ipsilateral problems. That's the main point to take away from that. We have a thing called the homunculus. So when you have sensor information that comes up for conscious awareness, there's actually a somatotopic arrangement of where different parts of our body end up in our cerebrum. And there's a very specific part called the postcentral gyrus, which we're going to identify on our real brains here. This little area of the brain. If we look at it, you can actually lay out a map of the body, and when those pathways are coming up, remember I said leg comes over here? So let's see, the leg tends to be kind of coming, and the knee and the foot. The foot is actually on the medial surface of the cerebrum, so your other half of your cerebrum is over here. Then your hand is out here, your face is down here, jaw, tongue, intra-abdominal area. So you can actually see your leg is more up here, and then your hand. And what they're showing here, the relative size of this representation shows you how much surface area or neurons are represented in that homunculus. So you see that how the thumb is big because you have a lot of neurons just dedicated that are in a lot of space or surface area taken up by your thumb because there's lots of sensory receptors. You use your thumb for manipulating objects, so it's got actually a larger region in that homunculus devoted to just the thumb. Look at your lips. Lips and mouth and tongue are very rich with sensory, and when you're manipulating or trying to speak, and you need that feedback and that sensory information coming in. So a large part of your cerebral area for sensation is taken up by hand and face. A smaller part is that actually you don't have a lot of receptors in your trunk and your feet. It's not as rich, you know. There's not as much density of receptors because you're not having to manipulate fine objects or really feel, you know, the fine ridges of something and those kind of things. So you have a less representation. It takes up less space. Motor. So that was sensory. Okay. We had our proprioception, which was the dorsal column, medial meniscus, pain and temperature, spinal thalamic / anterolateral system, and spinal cerebellar, unconscious proprioception. Now motor pathways. Just like we have a certain region in our cerebrum that's where the sensory information goes to eventually, we have a certain region in our cerebrum where motor signals start. So we're going to start at the cerebrum and work our way down, okay? We have these regions in our cerebrum, which we'll identify in the real brains. We have the primary motor area, which, let me change the color of this. Primary motor area is this area right here, okay? Right in front of the central sulcus. And then we have a supplementary motor area, a premotor area, and an area right here just in front and low called the Broca's area, okay? Primary motor area is for movement, execution, and fractionation. More than 50% of that area is dedicated to hand function and speech. Just like a lot of sensory space or real estate is associated with the hand and face, so is a lot of motor function. Trying to form words, trying to manipulate objects and work all those little muscles in your hand takes a lot of processing power, okay? The supplementary motor area, this area right up here, is primarily active especially prior to bimanual tasks like buttoning your shirt or opening a jar, things that require, you know, use of limbs together, okay? And it stores some of those patterns and sequences together. And it's also associated with sequential movements that require you have to do one sequence before the next, you know, and it stores those little programs that are these basic programs for some of that. Premotor area is for generating patterns of movements, especially involving multiple joint muscles and groups, especially that are more pendular in nature, like a golf swing or something like it's more of a pattern of movements of, you know, some of those basic patterns are stored there. But like the primary motor area, number 4 there, if I just want to wiggle my one finger, or I want to do this very novel task with just my fingers, you know, that's where that's going to come from, that primary motor. Or I just want to wiggle my one toe, you know, or move my ankle just so. That's where the primary motor. Supplementary and premotor areas are more with pattern movements, bimanual, stored kind of repetitive things we do on a motor tasks. We already seen that. So yeah. Oh, I'm sorry, the 4, 6, and 6, those are Brodmann's numbers. So there's different regions of our brain. There's actually a numbering system, okay, that actually has the numbers. So those are Brodmann's numbers, okay, that are mainly associated with those areas. Thanks for pointing that out. These motor pathways, so once you get out of the cortex and the cerebrum where they start, and they start to go down into the brainstem and down to the spinal cord, they're kind of like the sensory pathways. We have ways of grouping these together a little bit. So we're going to just talk about the main ones. There's a lot more. So there's actually 11 different ones we could talk about, but you guys don't need to know that. We will grossly divide this into what's called the lateral activating pathways, the medial activating pathways. These are going to the body, arms, legs, okay? Just what it sounds like. Lateral activating pathways are going to like more distal type control of muscles. Lateral, you know, distal, lateral. So controlling, you know, fine movements, novel movements, fractionation of movements and so forth. The lateral cortical spinal tract. That's the one that most people know about. Yeah.
What do you mean by fractionation of movement?
Like being able to individually move my fingers independently of each other, and like as opposed to doing gross big movements of working limbs, so actually doing very fine fractionation of movements. So good. So being able to really control fine movements is these lateral activating pathways. The lateral corticospinal tract is the one that probably most people need to be aware of. And the lateral listen, look at the word lateral. It runs in the lateral part of the spinal cord once it gets down there. And it's starting in the cortex, cortico, and it's ending in the spinal cord, corticospinal. It's a lateral corticospinal. And because it starts in the cortex and ends in the spinal cord, you know it's a motor tract because it's going from the brain toward the spinal cord versus spinal thalamic is going from the spinal cord. That's a sensory pathway. This is an efferent pathway. The rubrospinal tract actually starts in the brainstem. Rubro is red. It starts in the red nucleus, but it's more for upper extremity flexion. It biases. We do a lot of stuff where we bring things to our mouth. Primates are climbing. They do a lot of upper extremity flexion. The rubrospinal tract is for flexion of the upper extremities, and don't worry too much about that, okay? Then we have the medial activating pathways. The medial activating pathways tend to be most of these pathways start in our brainstem. There is one that starts in the cerebrum, but these guys start in our brainstem, and they control more of our postural muscles. The muscles around our spine, our girdle muscles, the ones that are for setting our postural set and tone and respond to automatic like changes in posture, like to keep you upright, not fall over. Keep you sitting up without having to think about it. Controlling the abs, the core, that kind of stuff. The medial, it's like medial proximal muscles activating system, okay? And you'll see there's the medial corticospinal tract. There's actually, the lateral corticospinal tract, but there's a medial corticospinal tract that comes down the middle part of the spinal cord. It mainly goes to the muscles of the core and around the spine, okay? Then you have these things called the tecto, rubro. I should actually take rubro out of there, cross rubro off. That's not supposed to be in there. That's part of the lateral activating pathway. The tecto, reticulo, vestibulospinal tracts. Just group those all together and just know that those start in the brainstem. Those all start in the brainstem and go to control postural muscles. Okay? So anytime you're reading a book and it says the tectospinal tract, the reticulospinal tract, the vestibulospinal tracts, those are all just starting in the brainstem, different locations in the brainstem, and control postural movements and postural muscles. Okay? Lastly, we do have a pathway called the cortical bulbar tracts. So we do have some control from our cortex to cranial nerves. Like if I want to stick my tongue out, purposely, or I want to smile, or I want to do something like that, I'm controlling those muscles via the cortical bulbar tracts. The cortical bulbar tracts are just the white matter going from the cortex to the cranial nerves, okay? And the motor parts of the cranial nerves that move your face, tongue, jaw, neck, that kind of stuff. The bulb, the brainstem is also referred to as the bulb. The bulb, it's like a bulbar structure. It's kind of rounded and bulbous in nature. So that's what they mean by the bulbar structure, is referring to the brainstem. Cortical bulbar, cranial nerves, okay? Our motor pathways are crossed, right? Especially the lateral activating pathways and the cortical bulbar tracts. They have a high level of contralateral crossing. So right-sided brain problem, left-sided motor problem, okay? Right-sided brain problem, left-sided face problem, okay? The medial activating pathways, they're not quite, they have like more bilateral connections, and some are even ipsilateral. Don't worry too much about that, but because they're kind of controlling your core, kind of just keeping your core upright. This is just showing you the cortical spinal tract. Most of them cross over. Some of these cortical bulbar tracts end here at the different cranial nerves on their way down through the brainstem. And then the medial cortical spinal tract doesn't cross over. It goes down the medial or the medial part of the spinal cord. The lateral cortical spinal tract that crosses over goes to the distal muscles. This guy, the medial cortical spinal tract, is going to more proximal trunk muscles. It doesn't cross over. Okay. Simple. Here's what I like about this is like all those pathways we just talked about in a simple diagram. Okay. This is showing you the division right here between brainstem and spinal cord. This is thalamus which will eventually take you up to the cortex right here. So if you look at pain and temperature right here, information comes in from one side of the body, it crosses over immediately, goes up the spinal thalamic tract through the brainstem to the thalamus, and then up to the cerebrum. Proprioception, conscious proprioception, comes in the spinal cord, stays on the same side for a little while as part of the dorsal columns, but crosses over right at the brainstem and then becomes the medial lemniscus up to the thalamus and the cortex for conscious awareness. Light touch, don't worry about it. It's actually, light touch is carried on both pathways, but that's why light touch is not a great way to assess your patient's sensation, because it's actually carried on both the dorsal columns and medial lemniscal systems, so you're not testing or not isolating the systems. It's better to test true proprioception and true pain and temperature. Here's unconscious proprioception. Comes in one side of the body, stays on the same side of the body as the spinocerebellar tract, and then stays and goes into the same side of the cerebellum. Ipsilateral. Doesn't cross over. And then here's just your major lateral cortical spinal tract. Starts in the motor cortex, crosses over. Remember in that decussation of the pyramids in the brainstem? Crosses over and goes down that lateral part of the spinal cord, eventually to the anterior horn, and then the lower motor neuron, which goes out to the muscle. So there's everything I just talked about in one little simple diagram. This is what works for me without having to memorize all the words. If I just know this kind of diagram, I can kind of see it and picture it, or be able to draw it out that way. Is helpful. Don't worry about that. This is just showing you how everything's layered in there. Here's your dorsal columns back here. This is, everything in blue is sensory pathways. Everything in red is motor. And believe it or not, that's how it's all squeezed in there. And it's actually layered in, lumbar, thoracic, cervical, different regions of your body are layered in as they come in to these pathways going up and down, okay? So that's all, you don't have to know anything other than that. I just wanted to get you an appreciation of how this is actually all put in there, okay? This big one right here is your lateral cortical spinal tract out here. This is your dorsal columns. This is your anterolateral system out here, okay? But cranial nerves, you have 12 of them, 12 pairs. The structures here in yellow are cranial nerves. So is this is the optic and the olfactory nerves. They're coming straight from the cerebrum, but all the rest come from the brainstem, okay? I'm not going to go through each one of those right now. You'll find your way to remember those. So these are, there's all kinds of different mnemonics and ways to memorize your cranial nerves and their function. The interesting thing, spinal nerves are always mixed nerves. Spinal nerves always have motor, sensory, and autonomic. Cranial nerves, some are mixed, some are pure motor, some are pure sensory, and some have those special functions like vestibular, vision, taste, those kind of things. So this just shows you if they have sensory, motor, both, what their primary function is. That's all this is showing you. Here's one cranial nerve mnemonic that you can use. Some people use this one here to memorize the different ways of so here you have the actual nerve and its name. Here's a mnemonic you can use to memorize that, and then some people use then this mnemonic. So the mnemonic on old Olympus towering tops, a Finn and German viewed some hops. That gives you the nerve, cranial nerve 1 through 12, and gives you the first letter of the name of each of those nerves if you look across to each of these. Then if you want to know whether it's sensory, motor, or both, you say this mnemonic. Some say marry, some say marry money, but my brother says bad business, marry money. Says bad business, marry money. There's other ones, that's pretty terrible. There's other ones that are better than that. I cannot tell you the one I learned because it's not G. It's definitely not G-rated. And now I'm mad because I can never get rid of it. Those mnemonics really do work because I can never get rid of this filthy mnemonic. So email me if you really want. No, I don't want to. I don't want to spread that one. So I'm sure you guys will come across it. All right, we're going to skip a little bit of this, and we're going to go straight to getting brains out. Any questions? Covered a lot today. I don't expect you to know all this today. This is just like to start getting you thinking about neuro. So when you come across it again later this week or next week, I'll go, oh yeah, I remember he said something about that, and you can go back to this and find it, or you can go back to my lecture and you can slow this down. So I need somebody from there's 7 specimens up here. How many tables do we have? 5 tables. So you're welcome there may be a table that wants more than 1 specimen, or there's actually a couple crappy specimens up here. Some of these are getting kind of used and abused, and some of them are a little ripe. And they're a little soft, and because they just didn't get the right quite amount of preservative. But these are cleaner than your cell phones are. So I promise you'll get more sick from somebody else's cell phone than you will from one of these brains. There's gloves up here. Send a representative from the table to bring at least 1 to your table, and bring some gloves. Not everybody has to handle it. I've got pointers in there, so if somebody can just be like pointing out structures, and I can, kind of make sure. And I'm going to draw the structures on here while you're going through it. Then if you raise your hand, I'll confirm a structure, or if you have a question about it. So somebody come up here, grab. If you want to touch the brain, come up and get some gloves so you can touch the brain. Or if you'd rather just have somebody be pointing to it and you can look, that's fine too. All right, what did I do? Pen. That's what I need, my little pen there. If you notice yours is pretty chewed up, you just can come up and grab a different one. If you can kind of, what's that? Rats? I hope not. Yeah, that one's pretty chewed up. This is a pretty good specimen right there. That's a nice one actually. If somebody had, there's a good specimen right here. If somebody's got one that looks a little chewed up, is yours kind of soggy? That's actually not a bad specimen. That's an okay one. That one's a little chewed up. Let me see. I think you guys got most of the okay ones. All right, let's walk you through the major surface anatomy here in the next half hour. Okay, I just had my pen, and now I just set it down again, my little drawing device. What did I do with that? Oh, all right. So we're going to go through each of the structures that are on here. The first one you cannot find. It's called the cerebral longitudinal fissure. We cut through, that's the thing that divides the 2 hemispheres. So we've cut through that. So you're looking at, we're going to start on the outside or lateral surface here. So we cut right down through the cerebral longitudinal fissure. So now we're going to find this first big structure called the lateral Sylvian fissure right here. The lateral fissure or Sylvian's fissure. That divides the temporal lobe from the frontal and parietal lobe area there. And if your brain has got enough, tissue structure, you can kind of peer in there and see some of that insular lobe. If you pull it apart gently, you'll see a few gyri and sulci down in there. That's going to be your insular lobe. That's more autonomic function, limbic function down in there. Okay. Now we're going to find the central sulcus. Everybody's brain is just a little different. In this particular specimen, this is the central sulcus. One way to know if you've got the central sulcus is the central sulcus tends to come over onto the medial surface, and just a little ways, it comes like, maybe an inch or a quarter of an inch, and it tends to dive posterior a little bit. So you follow it up over the top ridge, and it tends to then dive a little bit posterior, and that's usually how you know you're on the central sulcus. So it tends to dive kind of backwards like that and end pretty quickly after you go over the top ridge of the cerebrum. Did you find it?
Is this it? Is this one?
Yep, that is it. Good.
Is this one or is it that one?
Let me see here. Turn it this way. It's that one right there. Yep. It's kind of diving back that way.
It's just
Yep.
so straight.
Everybody find their central sulcus? Or think they found it pretty good? Your central?
Confirm ours. We're not positive.
Yep, that is it. And see how it kind of angles back
Yeah.
this way.
Well, this one kind of did too, and it's only.
It didn't go all the way.
Yeah, this one goes
Oh,
all the
okay.
way a little farther. But yeah. All right, so once you have your central sulcus, jump one sulcus forward to the precentral sulcus. So on this particular brain, it's this one right here. And sometimes you have to jump over a gyrus, but sometimes you have really good ones and it extends nice and cleanly all the way up. And this one actually kind of has two components to it that kind of join together, so it's not a perfect precentral sulcus. So we found the central sulcus. Now we jump one forward to precentral sulcus. Now jump one backwards from the central sulcus and find the postcentral. This particular picture's got a kind of a crappy postcentral sulcus. I actually have to jump over a gyrus here and connect the dots. Most of yours have better postcentral sulcuses. You guys feel pretty good on those? So now what you've done is you've defined the borders of some gyri, okay? So you define the borders of that's in front of the central sulcus, or between the central and precentral sulcus, is the precentral gyrus. Precentral gyrus. That is the area that is that primary motor strip. That's that Brodmann's area number 4, primary motor, that precentral gyrus, that is motor. So if you have damage there, you're going to have a motor problem. If the damage is down to this area down lower, okay, that's hand and face. So hand and face is down lower. As you get up towards the top and crest just over the edge, you've got more leg and trunk, okay? So this, big part of this lower region here is hand and face, okay, for motor. That's also true for sensory, which our primary sensory is the postcentral gyrus. So that gyrus just behind the central sulcus is the postcentral gyrus. That's all your primary sensory. And the homunculuses look very similar. They're almost identical. So there's a slight variation, but hand and face are lower on that homunculus. Trunk and leg are up more towards the top and lap over the medial surface just a little bit too. Okay. It just so happens that. Oh, let me draw this back out again. So like the most common stroke you can have is a middle cerebral artery stroke. Just the way that the internal carotid goes into the middle cerebral artery, like if a clot busts off, it tends to go into the middle cerebral artery. The middle cerebral artery tends to supply this particular region of your cerebrum. So it tends to wipe out motor and sensory mainly from the hand and face, not as bad from the leg, which is lucky for me as a physical therapist because the occupational therapy person has a lot harder trouble because their arm and face and speech therapy are tend to be more involved. Their leg might be a little involved, but they usually can get up and still walk, and I can work with that and those kind of things. But Preoccipital notch. If you come, if you have a you should all have a cerebellum, I think, left on your brains. Just above the cerebellum, about 2/3 of the way back, there's a little kind of a notch right here in the bottom of the cerebrum. There's a little, you almost have to imagine it sometimes. Some people's notches are more prominent than others. That's called the preoccipital notch. This just helps us know where the occipital lobe begins, okay? So if you kind of draw a line up from that, that kind of is what divides, separates you into your occipital lobe, okay? So here's our lateral sylvian fissure, central sulcus. Now if we just take our lateral sylvian fissure and just connect the lines, or extend the line or dots out, now we've divided ourselves into our 4 lobes. So here's frontal up here, here's parietal, temporal, and occipital back here. Okay, so that's kind of how you can kind of divide into your 4 lobes. Here in your temporal lobe, there's a gyrus right here. This whole region right here is the superior temporal gyrus. That's where your initial auditory information comes in. So when you actually process the actual initial sounds in a little region right there called the primary auditory area. Then out and around here, this is where you actually detect the pitch, the loudness, but then out here in the secondary area is where you decide if it's, you categorize it. You decide if is that a bell, or is that somebody talking to me, or you start to make sense of what that sound is actually giving it some meaning. Okay? Before you actually send it up to your parietal lobe to actually give it true meaning and identify what the meaning of that is. So that's the lateral surface. So we're just going to, that's a quick overview of the lateral surface. Now the medial surface. A few more things to look at here, a little more intimidating. Let's start out with that big thing in the middle right here called the corpus callosum. Those are all axons that are crossing, going from one side of the brain to the other. They're connecting corresponding frontal, parietal, occipital lobes from one side to the other. So you can communicate and send signals between each side of your brain. Oops. It's got some different regions to it. It's got a beak or a rostrum right here, the little tip right here that's kind of curled underneath there. It's got this region right here, which is the bend, which is called the genu. Okay, so that's the genu. Genu means bend. Then you have the body is all this region here, this kind of more straight section. And then this back part is called the splenium. Okay, so it's got 4 different parts to it associated with the corpus callosum. Diencephalon. Some of yours is kind of a little bit roughed up, but I'm going to outline the diencephalon is this whole region here. And then if I just kind of follow that around like that, so the egg-like structure here is the thalamus. This region here is the hypothalamus. Some of you will have this little bump right here called intrathalamic adhesion. A lot of you won't. It's been cut off or just they didn't have one. Your diencephalon, this is your third ventricle too, right here. As long as you have another one next to it, that little space in between is your third ventricle, okay? But you got to have 2. You got to have another diencephalon next to it, that little space there to have your third ventricle. There's a little bundle of white axons right here called the anterior commissure. That's kind of like the corpus callosum, but it connects the 2. There's a temporal lobe here. It actually goes down and connects one temporal lobe to the other temporal lobe. There's some fibers that run from temporal lobe to temporal lobe on each side, called the anterior commissure. So that's the anterior commissure. Preoccipital sulcus. There's a really big sulcus right here called the preoccipital sulcus. This is the occipital lobe. This is your parietal lobe here. Okay. Frontal lobe is about right there where it stops. Here's your central sulcus right up there. But this is the pre-occipital sulcus, is that big one right there. Okay. Then we can take our occipital lobe and divide it into a couple parts. There's this thing called the calcarine sulcus right there. There's a fairly decent sulcus right there called the calcarine sulcus. And that divides us into this little thing right here called the lingual gyrus. Lingual means tongue-like, and that kind of looks like a tongue, I guess. And then this up here is called the cuneus. This region above is called the cuneus. Right on this inferior border of the cuneus and superior border of the lingual gyrus, that's primary vision. So when your initial visual signals come to your brain, they go right to the little edges of that calcarine sulcus on each side, and your visual stuff comes into your cerebrum on the opposite side and upside down. So this little region right here is actually bringing in information from the lower half of your left visual field. And this is the right side of the brain. It's on the upper part. And this is vice versa. This region right here is bringing in information from the upper half of your left visual field. And then your brain puts it all together and makes sense of it. So it doesn't necessarily matter where it comes in. Eventually it gets put back together to give you the appropriate picture you're looking for. Cingulate gyrus. C, cingulate is kind of C, it's C-shaped. So that is the cingulate gyrus right there. And the sulcus above it is called the cingulate sulcus. So the sulcus right here that defines the top part of that is called the cingulate sulcus. The cingulate gyrus and sulcus, these are parts of the limbic system. This has to do with emotions, okay? Emotional responses and so forth. There's a sulcus right above the corpus callosum called the callosal sulcus. Makes sense, right above the corpus callosum. So the cingulate gyrus right here is defined by the callosal sulcus and below it and the cingulate sulcus above it. Okay. Oh, some of you do not have this. Some of you still have a little bit of an optic nerve coming off here. Optic nerve, and there's a little bit of a white area right here called the optic chiasm. Part of your visual information crosses there. It goes to the opposite side. So there's a chiasm as a crossing point. And so some of you, depending on how well we cut it, have a little optic chiasm with one of the optic nerves, tag end of an optic nerve coming off. Okay. Here is the midbrain right here. This is all midbrain. There's the cerebral aqueduct. There's a little aqueduct going from the third ventricle down into this fourth ventricle right here. That space is the fourth ventricle. And then eventually that stuff gets out from below the cerebellum. Here's our pons, this whole region here, and this rounded part is the basilar pons. And then down here is medulla. Below that is the medulla. Some of you do not, a lot of your medullas got cut off because we didn't quite get the cut low enough when we were taking the brain out. So, but all of you, most of you have a pons and a midbrain there. Some of you will be able to, most of you can take your little pointers and you can, this particular specimen has this little membrane here called the septum pellucidum that covers over between the two ventricles. But most of you guys have got this cut away, so there should be a space there, and you can like look in there, and you may be able to see a little bit of leftover, if it hasn't been dug out, of some little choroid plexus somewhere down in the floor of this, deep in there. You might see this little granulated little tissue, looks like maybe raw hamburger or something in there, little pieces of it that are the choroid plexus that produce the cerebral spinal fluid. There's a little hole right here. Some of you still have this little piece. There's a little piece right here of tissue called the fornix. Some of you still have that. There's a hole right there. And if you poke your pointer up through that hole, you'll see it come out inside of the lateral ventricle. That's the intraventricular foramen. Between the ventricles, it's the hole that allows you, the lateral ventricles, to dump into the third ventricle. Some of you can stick your little pointer up through that little hole and see it come through there. And then, take a look at your cerebellum back here. This right here, this like smaller 1/3 portion, that's the anterior lobe. And then this big 2/3 back here is the posterior lobe of the cerebellum. We're not going to worry about this.